Introduction to Biosensor for Diagnostics. Diagnostic biosensors are generally used for the diagnosis of disease, monitoring of disease, drug discovery, and detection of disease causing pathogen. For the designing and fabrication of diagnostic biosensors, there are 5 important factors which are needed to be considered. The first is the nature of the analyte or the target molecule. It means what type of molecules we are going to detect by using biosensor technology. The second is the sample matrix. Sample matrix means the sample in which our target molecule or analyte is present. We need to consider the nature of the sample matrix. Is it simple or complex? Number 3, the type of biorecognition element. It is very important to consider the type of biorecognition element in a diagnostic biosensor because the performance of diagnostic biosensor is heavily relied on the bioreceptor molecules. There are different types of bioreceptor molecules, for example, enzymes, DNA, different types of hormone or biological cells. Fourth point is the transduction process. We know that when a bioreceptor molecule binds with its target molecule, a signal is produced. This signal is detected by a transducer. There are different types of transducer, for example, optical transducer, electrochemical transducer, magnetic transducer. We have to decide about the transducer as well because transducer is the main physical component of the biosensor that is going to detect the signal and amplify it. The performance of the diagnostic biosensor is again heavily relied on the transducer or the transduction technique which we are employed in the fabrication of the diagnostic biosensor. Fifth point is the material and the methods that are used for the immobilization of the bioreceptor molecule on the transducer. This factor is also very important. In this module, we will only discuss the type of analyte or the nature of analyte. In diagnostic biosensor, what do? We basically determine the biomarker. What is a biomarker? Biomarker is a biological element, and this biological element appears to a particular conditions. This molecule could be, this element could be determined and evaluated. It is produced in response to a normal biological process or a pathogenic process or a pharmacological process. And the other thing is the detection of biomarker is very useful for the early detection of the disease. And several diseases like cancer could be treated if we can diagnose these diseases in time. Now, what are the examples of these biomarkers? The examples of biomarker includes a pulse, blood pressure, body temperature, blood glucose level, basic chemistries of body fluids, and complex laboratory test of the body fluids. All these are considered as a biomarkers. These biomarkers could be physical in nature, they could be molecular in nature, or they could be there could be other types of the biomarkers. Broadly, we can classify biomarkers into 2 types. One is the qualitative biomarkers and other is the quantitative biomarkers. Actually, biomarkers provide us the information that is as a laboratory parameter. And this laboratory parameter is used by the physician or doctors to make a decision about the diagnosis and the selecting a course of treatment for the patient. So, what is qualitative biomarker and a quantitative biomarker? What is the difference between them? These are both biomarkers. In case of qualitative biomarkers, the pathogen process detection within it provide us the answer as yes and no. And in case of qualitative biomarker, we get the answer pathogenic process detection with a threshold effect. It means the level of that particular biomarker, that what is the level of that particular biomarker, and it could be related to the disease condition. Now, the types of biomarker. There are generally 7 types of biomarker. First is susceptibility or risk biomarker. They identify the risk of disease or exposure sensitivity. Second type is diagnostic biomarkers. Diagnostic biomarkers diagnose the conditions of interest, disease, and subset of disease. Next is the monitoring biomarkers, and these basically monitoring biomarkers detect changes in the degree of disease or the extent of disease. The fourth type of biomarkers is prognostic biomarkers. They identify the likelihood of the clinical event and they indicate disease recurrence and prognosis. The fifth type of biomarker is a predictive biomarker. This biomarker predicts the individual who will be favorably or unfavorably could get the disease, and it also indicates toxicity or assess safety. Number 6 is the responsive biomarker. It shows the biological response occurred in an individual for a drug. Number 7 is the safety biomarker that indicates the extent of toxicity related to the exposure of exposure or intervention of the drug. Now, in case of a disease, several types of molecules are released into the blood circulation or they are present in the body fluid. So what type of biomarker which we are going to select? So an ideal biomarker should be cost effective. It should be correlated with the disease activity. It should be easy to perform. It means it should be convenient to identify it. It should be specific, useful to follow up, and useful to assess the severity. It means the information that will be generated by studying the biomarker should be very useful. Now, in a laboratory or in medicine, what are the biomarkers? This slide will give you an idea about the biomarkers, how these biomarkers are useful in medicine. First, you can see we are using blood as a sample, and this blood sample is tested for different types of biomarkers. It could be a DNA, it could be a protein, it could be an antigen, or it could be any type of cell. And all these samples react with a bioreceptor element. When they react with the bioreceptor element, once the target molecule is detected, there is a signal generated. This signal is picked by the transducer and is converted into a readable signal. This readable signal is very useful. It can provide us the information about the diagnosis, about the screening, prognosis of disease, and other important factors that are related to the medicine. So that is how these biomarkers are useful. Now, the information generated by these biomarkers is equally useful for a scientist and a clinician or a medical doctor. For a medical doctor, it is helpful for the early diagnosis of disease. It is also helpful for the evaluation of drug efficacy, patient care decisions, drug toxicity assessment, and disease prognosis. And for a scientist, the scientist can use this information in bioinformatics and computational biology, genomics, epigenomics, proteomics, system biology, and structural biology. So a huge amount of information could be generated by studying the biomarkers. Now, I will give you an example in which the biomarker, a specific biomarker, is helpful in diagnosing the breast cancer. This is human epidermal growth factor receptor HER2. This protein in normal cell is present in very little amount, but in case of cancerous cell, the amount of this HER protein is very high, and this protein could be detected through ELISA in blood circulations. Once this protein is detected, breast cancer could be diagnosed. Now, in summary, what we have learned so far, there are 5 different types of level at which the diagnostic biosensors could be focused, such as biomarkers, sample, bioreceptor, transducer, and immobilization layer. A biomarker is a signature molecule and it tells us about the pathological condition of a patient. An ideal biomarker is specific, informative, and cost effective. Detection of biomarker is helpful in early diagnosis of disease. So there are few related questions. What are the important factors required for designing a diagnostic biosensor? Sorry. What is a biomarker? Give different types of biomarkers. How biomarkers are useful in diagnostic and what are their benefits?